So we start in the seminiferous tubules. This is where spermatogenesis occurs within the cytoplasm of the cystinocyte or Sertoli cell. So FSH is going to target these cells to allow for spermatogenesis. The first step is where our stem cells or spermatogonium undergo mitosis, and we are going to allow for the creation of a type A daughter cell, which will stay at the basal lamina and it will continue being a stem cell, and a type B daughter cell. And this type B daughter cell is going to begin meiosis. And there is a separation between the type B daughter cells and the primary spermatocytes called the blood-testes barrier. And that blood-testes barrier prevents the immune system from not recognizing these sperm cells, from recognizing them as foreign and trying to kill them. So our type B daughter cells begin meiosis and they become primary spermatocytes. Primary spermatocytes complete meiosis 1 to become secondary spermatocytes. Secondary spermatocytes complete meiosis 2 to become early spermatids. Early spermatids are going to basically just do a costume change to become late spermatids, and ultimately fully functioning sperm or spermatozoa. And this costume change where spermatids become sperm or spermatozoa is called spermiogenesis. Mitosis plus meiosis plus spermiogenesis equals spermatogenesis. So this is all occurring within the seminiferous tubules, specifically in the cytoplasm of a cystinocyte. We also have Leydig or interstitial cells there targeted by the hormone LH from our anterior pituitary, and that's going to make testosterone, our male sex hormone. So we've made these sperm cells. They have a head with an acrosome on it containing the DNA, and the acrosome allows the sperm to penetrate the zona pellucida around the oocyte. Midpiece contains mitochondria, which is how we're going to convert fructose into ATP, which is energy for this cell, and the tail is for motility. So now this fully formed sperm cell is going to travel from the seminiferous tubules into the epididymis, both of which are in the testes. Then it's going to travel in the ductus deferens, which is within the spermatic cord. Ductus deferens will join up with the seminal vesicle, which will release its product, which is a fluid containing fructose. These will join together to form the ejaculatory duct, which is very, very short. So the ejaculatory duct will then lead into the prostatic urethra, which is within the prostate gland. The prostate is going to secrete a fluid containing an activating enzyme, which activates the sperm. We then travel through the pelvic floor muscles through the membranous urethra, which contains the bulbourethral glands right next to the urethra, which is going to secrete an alkaline fluid, which neutralizes acidic urine left in the urethra. It also neutralizes the female reproductive tract. We then travel into the spongy urethra, which is found in the corpus spongiosum, which is our erectile tissue in the penis. The corpus spongiosum and the corpora cavernosa are two types of erectile tissue. Erection will be initiated by the parasympathetic nervous system. Ejaculation will be elicited by the sympathetic nervous system. And so ultimately, sperm will then leave the body after ejaculation.